Hello and welcome to Nikolai's Genetics Lessons, and in this video I'm going to talk about leaky scanning. We can find this mechanism of diversification of proteins production in both eukaryotes and viruses. And for example, just 25 years ago, scientists and geneticists were thinking that because we humans have over 100,000 proteins, that means that we should have over 100,000 genes in order to code for all those proteins. But it's turned out, after our genome have been sequenced, that number of genes only about 23,000. And we have all this diversity of proteins due to alternative splicing of our genes, that one gene can produce 3, 5, or even more different variants of the same proteins. And leaky scanning is similar to this mechanism that produce diversity, as I said, in both eukaryotes and viruses. So, for example, if one virus has 8 genes, this doesn't mean that this virus would produce only 8 proteins. This virus can produce 10 or 12 different proteins, but not due to alternative splicing, but due to leaking scanning. So imagine that we have messenger RNA that is produced from gene of the virus, and we also would have 5' cap here, and we also would have poly A tail here. And now this messenger RNA can be translated into protein. And how this mechanism works: first of all, small ribosomal subunit would be attached at this place on the messenger RNA, and then it would start moving down the stream in order to find the sequence that is AUG, that code for the methionine, and also would be a start codon. And if in prokaryotes we have such sequence that we call Shine-Tilgarna, to which small ribosomal subunit would attach, in viruses we have other sequence that we call Kozak sequence, and this AUG would be a part of this sequence. So when this small ribosomal subunit would reach this sequence, so it would stop and ribosomal complex would be assembled. So once again, here is going to be small ribosomal subunit, and how this small ribosomal subunit would recognize this sequence. Once again, this sequence just going to be a part of the Kozak sequence, so it's much bigger, and actually this is not just a protein, this small ribosomal subunit, but about 50% it is RNA and proteins associated with this RNA. And because this is single-stranded RNA, of course, it can find a match of this sequence and would stop. And next, the whole complex, ribosomal complex, including large subunit, ribosomal subunit, would be assembled. And as you remember, there are 3 sites here, 1, 2, and 3. So the first site would be A site, P site, and E site. And the first amino acid would go into the P site. From the textbooks, you probably know that the first site where amino acid would go would be A site, but actually the very first amino acid would go to the P site. and all the rest, of course, would go to the A site, and then to the P site, and to the E site, and would exit. So imagine now different variant that down the stream we have another AUG site, and as you know, in this case, when this ribosome would be assembled and would be moving down this messenger RNA, this AUG just basically means not start codon but methionine. So methionine would be attached here at the A site of the ribosome. But what would happen if this sequence here also would have a Kozak sequence? So let me use some color to designate this sequence. What if here we also would have this Kozak sequence? That means that this is also can be a start codon, and this is going to be, in this case, alternative attachment site for the ribosome. For a small ribosomal subunit, and then large ribosomal subunit also would be attached, and this is going to be a second alternative site for the beginning of the translation of protein, and this is going to be first variant. So that also means that we can get two variants of the proteins. One protein would start being translated from this side, and the second would be shorter and would start from alternative site of the attachment for the small ribosomal subunit. So we would have basically the same protein, and of course then protein would be shaped into its final shape. So it's going to be basically the same protein, but one would be a shorter version of the other. So probably they would have the same function but slightly different properties, and there can be many such variants. Say there can be even third or fourth variant of the same protein, and this is a mechanism of the diversification of the proteins in viruses that we call Leaky scanning. But this is not the only variant. This is so-called variant number 1, and there is also another variants exist. Variant number 2 would be a mechanism when we would have the same messenger RNA, just like in example above. Once again, poly A tail would be added here, and also 5' cap would be added here. And once again, imagine that somewhere here we have beginning of the translation AUG site, and we would have Kozak sequence, and AUG just would be a part of this Kozak sequence. And as you know, there are different variants how the same messenger RNA can be read. For example, just for simplicity, I would add another nucleotide here that is going to be U, and then again A, U, G, then A, and A, U, G, and say U, A. So because ribosome would move along this messenger RNA and would read the sequence as trinucleotides, we would have in the first variant when a ribosomal complex would be assembled here, we would have the following trinucleotides. So this is going to be the first one, here is going to be the second one, the third one, the fourth one, the fifth one, and so on. But now let's imagine that our small subunit ribosome would attach here, and as you see, in this case we are not going to have the shortened version of the same protein, just like in the first example, but actually the reading frame itself would shift here, and here's going what happen. So this is going to be the first trinucleotide, this is going to be the second, and this is going to be the third, and so on. So as you see, the sequence of the codons would be different. Here, sequence of the first codon AUG, then UAU, then GAA. In this scenario, we would have sequence AUG here, AAA here, UGU here. So the same messenger RNA, if would have start codon, which is going to be in the different reading frame, then this would produce 2 or 3 different, completely different proteins. Because if we have AAA sequence here, this would code for one variant of the amino acid, but if we would have GAA, of course we would have a completely different amino acid here. So this sequence would produce one sequence of the amino acid, and this variant would produce different sequence of the amino acid. So this is going to be completely different proteins. And as you see, total three possible variants are possible here of the 3 different reading frames. And this would give us, once again, 3 alternative variants how the same messenger RNA can be read. And of course, also due to Leaky scanning, not just 3 different variants of the proteins the same protein can produce, but also there can be different start codon on each sequence. But it is very unlikely that some sequence would have some meaning in one reading frame and also would have meaning in other reading frame and in the third reading frame. So the most variants of the proteins diversity that can be produced by single gene or messenger RNA, we would see that described here as a first variant of the Leaky scanning. And when you would see that, for example, some virus like influenza virus A, which has 8 genes, may produce 10 different proteins, in most cases this is going to be this variant when the same protein may have different lengths and different N-terminus. So each protein, as you remember, has N-terminus and C-terminus. So the C-terminus would be the same, but N-terminus would be different in each such protein. Just like if you would take a look at this messenger RNA, each messenger RNA has 5' end, which is beginning, and 3' end, which is the ending of the messenger RNA. Each protein also has N-terminus and C-terminus. One more time, the first variant gives us different variants of the same protein, just maybe different variants of the length of the same protein, because these start codons would be in the same reading frame. But in this variant, we see that start codons in different reading frames and would give rise to completely different proteins coded by the same messenger RNA. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. Share this video with your classmates and see you in the next video. Goodbye.